At the onset, I want to thank Professor Anuj Maheshwari, sir, and Dr. Alok Modi for giving me this opportunity to share my thoughts on this very interesting topic amongst this august audience. So today we will discuss about PKPD in CKD and how we can utilize artificial intelligence to optimize drug therapy in chronic kidney disease patient. Hope my screen is visible and it is changing. So when we talk about any therapeutics, as prescriber, it is our duty to balance between risk versus benefit. Risk associated with any specific treatment should be balanced with the benefit that would be provided by that particular treatment. And for that, we need to understand two important concepts: the relationship between drug concentration and time, along with the drug concentration and its effect. And applying that knowledge, we should understand how our treatment regimen can be provided in therapeutic range. It should not go above, that is supratherapeutic range, or it should not go below, that is the subtherapeutic range. And this interesting concept of pharmacokinetic pharmacodynamics ultimately help us to understand whether drug that we are using that could be efficacious or that could produce some toxicity or some subtherapeutic response. So as we understand, so I think this is visible. When drug is administered, there are few important characteristics that actually governs the drug concentration in systemic circulation. And sometimes we focus for some locally acting drug, like for an example, inhalational corticosteroid, the site of action is lungs. So for that purpose, the pharmacokinetics is important. Then how our genetic polymorphism influences the drug response, that is also critical, and that is why we need to apply the principles of pharmacogenomics. And finally, the pharmacodynamics at the level of receptors or ion channels or enzymes or immune system like T cells or antigen present in cell, how the drug is acting. That governs the clinical response and ultimately determine the clinical efficacy. So starting with PK, pharmacokinetics, this is the study of the time course of a drug's movement through the body. We need to understand what the body does or with drug. So absorption, distribution, metabolism, and elimination, these are four important parameters for pharmacokinetics, what the body drugs to the drug. And as we are talking about ADME, absorption, distribution, metabolism, and elimination of a drug, we need to understand this interrelationship and concentration at its sites of action. Especially when we talk about therapeutic site of action, we need to understand the drug should reach there at the receptor level in a free form. And that is most important because, again, they will also reach to the unwanted site of action, and that can cause adverse effect. So everything that determine inside our compartment, that is the central compartment, how the free drug concentration is available there. So in CKD, we know the protein concentration, the albumin concentration goes down, and which is a very critical factor because the drugs which are mostly protein bound, they will not get that protein to be bound with, and ultimately the free drug concentration will be more, and that may lead to certain toxicity. After that, if we concentrate in this position, the excretion part, the elimination of drug, that will also hamper when we are talking about our chronic kidney disease patient. Uh, Even the metabolism, there is some changes that happened in chronic kidney disease. So absorptions, they are generally not much well quantified. Sometimes it increases or decreases. Volume of distribution also, there is generally no change, but sometimes it increases. Metabolism decreases due to decreased action of different cytochrome enzymes. And obviously the elimination through kidney, that is decreased. But non-kidney elimination, they are also poorly quantified, and there is a chance of possible decrease in that. So when we talk about pharmacokinetics, this chart, this curve, the x-axis with time post-ingestion and y-axis with plasma concentration is an important part. For an oral drug, the absorption will take some time. So this is an absorption lag time. Then the concentration of the drugs increases and it reaches the maximum concentration. And to reach the maximum concentration, the time taken, that is the Tmax. And when we talk about 50% decrease in concentration, like from here to here, the time taken, that is the elimination half-life. So these are again important pharmacokinetic parameters that we need to understand when we are talking changes in CKD. So clearance is something which is mainly affected in chronic kidney disease patient. We need to understand clearance is not only depends on kidney, it also depends on hepatic and some other organs that include skin. So when we talk about some drugs which is 100% cleared by kidney, like in this blue line, this had been shown. So therefore, it is predicted that a 50% decrease in GFR will also correlate with 50% decrease in total clearance. So that is for the drug B. But who are under that drug B category? This is the black line. They are having no kidney clearance, so non-kidney clearance. So they are not much affected when there is chronic kidney disease. There are certain exceptions too. But when we talk about those drugs which has both kidney and non-kidney clearance, that need to be kept in a special consideration. So for an example, if we talk about antibiotics like beta-lactams or aminoglycosides, they have only kidney clearance. Now for macrolides, they are non-kidney clearance only. But for fluoroquinolones, they have both kidney and non-kidney clearance. There are certain fluoroquinolones like moxifloxacin, which has very less kidney clearance. But when we talk about anticoagulants like dabigatran, they have kidney clearance only, whereas warfarin has non-kidney clearance and rivaroxaban has both kidney and non-kidney clearance. Like when you talk about beta blockers like atenolol, they have kidney clearance only. Metoprolol, they have non-kidney clearance only, mostly hepatic. And when we talk about bisoprolol, they had both kidney and non-kidney clearance. We were talking about CYP cytochrome enzymes. So the studies had shown when we talk about chronic kidney disease, declining kidney function, the drug clearance by metabolism also can be decreased. So these are some prototype drug which are being tagged with some specific enzyme. Now, if we can see this particular study, how each of those enzymes starting from CYP1A2 to CYP2C8 to C19, all those metabolism had been decreased. So now if we can corroborate that results with some specific metabolizing drugs, for an example, warfarin, if there is decreased clearance by CYP2C9, the warfarin concentration will be increased. So enzymatic metabolism decreased, that means there will be decreased clearance. So ultimately, what should be our goal? We need to decrease the maintenance dose, or we need to increase the interval of dosing. So decrease the frequency of dosing. That means if it is a twice daily drug we need to give it once daily. So this we need to remember. There are certain criteria for dose adjustment according to the pharmacokinetic parameters. These are important to note. And when we talk about some specific threshold, if there is pharmacokinetic parameters changes less than 30%, I think there is no adjustment is much required for that particular drug. But if it is more than that, then we need to be cautious, especially when the area under curve for any particular drug increased by 50 to 100%, or the drug having some narrow therapeutic range, for an example, theophylline, we need to be very cautious. We need to consider the decrease in maintenance dose or decrease in the frequency of dosing. General dictum is if the clearance decrease less than 50%, then for a particular drug, then that is not much affected with deteriorating kidney function. So dose adjustment generally unnecessary if less than 30% of dose is excreted by kidney. But for some hepatically metabolized drug, we need to understand the drug could be metabolized to certain active or toxic metabolites, and they are renally excreted. For an example, morphine to morphine 6 glucuronide, mycophenolate to mycophenolic acid glucuronide. So that is the time we need to be very cautious. Why we are talking about this all in presence of AI? Artificial intelligence, if we can put this data, that they can help us a lot. Apart from that, these are few important considerations, how we should behave while changing the dose in presence of chronic kidney disease. After pharmacokinetic, we need to understand the pharmacodynamics also changes. And here we must appreciate the pharmacodynamics is mostly following the pharmacokinetic changes. Here, this is a normal situation, but here it is a deteriorating kidney function. We can see the changes in the curve, both changes in the concentration and the effect, and how all those pharmacodynamics are mostly same, but due to change in the half-life because of presence of chronic kidney disease, that half-life increased, doubled, the TET50 also doubled, and therefore we must appreciate the need of this consideration of pharmacodynamics. There are certain factors that influence the clinical pharmacologic drug response as measured by onset of action, intensity, duration of effect, and these factors act primarily by affecting the drug concentration at the receptor site. So when we talk about drug dose, drug pharmacokinetics, receptor number, organ response to receptor activation, and certain counter-regulatory influences at the receptor, we need to appreciate this mechanism, and even presence of certain pathological processes including aging, acute or chronic illness, and kidney disease that can affect pharmacodynamics clinical response work. So there could be a decreased receptor number. So ultimately the Emax will be changed and the sensitivity CE50 will be changed. So decreased receptor binding, altered signal transduction that can be happened with chronic kidney disease. And therefore, the drugs that can interact and compete for similar receptors having multiple effects. It could be synergistic, antagonistic, or certain effect concentration correlation with Hill coefficient. So as we are talking about Hill coefficient, we can see if the Hill coefficient is same, but the C50 is increased. So what happened? We need to actually increase the dose of the drug to reach that normal effect. Similarly, if the Emax is halved, we need to increase the dose. And when H decreases, the Hill effect, the Hill coefficient decreases. So therefore, we need to again increase the dose of the drug to reach the desired effect. So this theory, the pharmacodynamic metrics, that also need to be considered while dosing in CKD patient. And of course, when we are talking about some surrogate markers, there are certain surrogate markers. For an example, if we talk about anti-infective agents like antiviral agents like silgosivir, we need to check that platelet count difference. We need to check that hematocrit difference, and that could help us as a surrogate marker for pharmacodynamic monitoring. We all understand artificial intelligence is designed to amplify. It augments human intelligence, not replace it. And there are different technologies. I think this need not to be discussed. Already we had a long discussion on what is artificial intelligence is, but now we need to understand how we can use this artificial intelligence, which is obviously intelligent, can recognize pattern, and could be used as a dynamic autonomous tool. So actually, we need to have a good patient outcome, and our treatment should targeted for a good patient-oriented outcome. So for that, in each step, starting from the patient phenotype here, some specific clinical, social, and genetic information to diagnosis, to the treatment decision making, what are the medication we are prescribing, how prescription is being filled, everything that those data, if we can put, they can tell us about certain possibility of adverse drug event. And if we also can go on with that that process, and we can collect the patient data, so that also help us to understand, to detect adverse drug event early, and ultimately we could have a large window of therapeutic response to react and to prevent bad outcome and to provide a good patient outcome. So when we are talking about drug-related problem management, there are certain already available part, like AI-driven alerts, like healthcare chatbots, how we can use precision dosing and remote telemedicine with this integration of artificial intelligence in drug-related problem management. This could significantly improve patient outcome. So these are certain innovative digital tools for medication safety and preventing drug overdose, like MedAware, MedStar Health, and the Pennsylvania Patient Safety Authority, MedEye, Prescriptimate, Lumio Medical, and IRODI, which are very essential. Generally, when we talk about medication safety, these are important points. We need to have that robust data, rigorous testing, and validation of those AI-powered tools. Then collaborative approach should be there between healthcare providers, data scientists, and technology experts. And we need to address ethical concerns like data privacy and algorithmic bias. So there should not be any specific algorithmic bias that these are essential to ensure the responsible and equitable use of artificial intelligence in medication management. These are few important devices that we are personally attached with. This is an cardiac autonomic neuropathy detection device. We are collecting data. We are putting them in the cloud, and we are prospectively doing the follow up to our patient. This is another device we are making, that is MedPer. They are adherence enhancing device. If we put 3 or 4 medications here and they have some drug-drug interactions, so they will give some specific alert too. Now coming to an interesting model, how the pharmacogenomics and the baseline drug development process can be incorporated and can tell us about future drug response. So with the help of artificial intelligence, we can understand what are the gene expression or mutation could influence the drug response. And another important study had been published. They had utilized artificial intelligence-based, physiology-based pharmacokinetic model, and they had they had predict some specific ADME parameters of an competitive potassium channel blockers. So that is vonoprazan. And we can see when the stimulated pharmacokinetic parameters become adjusted with this AI model, they are almost similar to the real life. So that is something interesting, and they can predict on other potassium competitive antagonist too. So there are another molecule they are preparing. Now they are utilizing this data on vonoprazan. So when we talk about CKD management, and that could be guided by artificial intelligence, there are certain biomarkers which can evaluate, we can prognosticate. There could be some alerting system, diagnostic assistance. Along with, we need to develop certain tools by incorporating this pharmacokinetic, pharmacodynamics characteristics with these AI models, and that can help us in precision medicine. So when we talk about future direction and research in AI, we need to conduct ongoing artificial intelligence research in nephrology and pharmacotherapy. We need to develop certain deep learning next-gen AI models, which we need to appreciate that there is a high potential for artificial intelligence to transform chronic kidney disease management. But for that, we need to have a proper collaboration between nephrologists, clinical pharmacologists, and artificial intelligence developer. So we should have a proper recap, basic concepts of pharmacokinetics, pharmacodynamics, that there are changes in chronic kidney disease. We need to understand the importance of dose adjustment in CKD patient, and artificial intelligence need to be considered as a valuable tool for improving dosing accuracy, modifying the patient outcome by having a more precision type of therapeutic application. We need to encourage to adopt artificial intelligence technologies for better clinical outcome. So once again, I want to thank Professor Anuj Maheshwari sir, who is my mentor, and Dr. Alok Modi for giving me this opportunity to share my thought on this important topic. Thank you very much.